So my name is Dallin Thewison,
I'm Kent Charrington.
And this is Logan Waite.
and today we're just going to be talking about Lesson 9 group problems. And so for the first problem, we're asked to just kind of draw the replication process of a DNA sequence. And so this is our awesome little picture. I'll just kind of point out the units. So this is a ORIC. It's just kind of the origin of replication, just kind of in that area. The DNA primase is right here. This is what's going to prime it's kind of like the primer, you could say, for the DNA polymerase 3 before it kind of attaches the DNA nucleotides. Let's see, the DNA helicase is right there. That's what's going to separate the DNA strands. We have the leading strand, which is right here, and these are the lagging strands. We could also consider the lagging strands as the Okazaki fragments. DNA polymerase 1, or like the proofreader, is behind the polymerase 3 alpha subunit, so this is right there. And then once again, these are the Okazaki fragments.
Okay, so for question 2, it's got just some fill in the blanks. So the DNA primase is the protein complex that synthesizes primers in eukaryotes. This protein promotes the separation of the AT-rich sequences within the ORIC, which is the DNA A. The epsilon subunit of DNA polymerase 3 contains proofreading activity. For part D, this protein synthesizes the leading strand in eukaryotes, which is the DNA polymerase epsilon. The TUS protein displaces DNA polymerase 3 from the chromosome to terminate replication. DNA polymerase 1 this protein has the 5' to 3' polymerase and 5' to 3' exonuclease activity. The DNA gyrase. This protein introduces negative supercoils into the DNA ahead of the replication fork. The ORC protein binds directly to the ARS elements. The SSBPs. This protein prevents DNA strands from annealing during replication. And DNA ligase is the enzyme that seals Okazaki fragments into a continuous strand of DNA. Alright, so for number 3, it asks us to list 5 enzymes that consume ATP during DNA replication. These include DNA primase, DNA gyrase, helicase, DNA A, MCM, and DNA ligase. For B, it asks for 5 enzymes that consume DATP, and these include DNA polymerase 1, DNA polymerase 3, DNA polymerase alpha and delta, and DNA polymerase epsilon. Part C, 5 enzymes that have 3' to 5' exonuclease activity, and those include DNA polymerase 1, 2, and 3, DNA polymerase delta, DNA polymerase epsilon. Indicate one enzyme that is involved in replication that uses DNA as a template to synthesize RNA. This would be DNA primase. And finally, indicate one enzyme that uses RNA as a template to synthesize DNA, which is telomerase.
Okay, so for question 4, it gives us a sequence of nucleotides. So it says suppose you're trying to synthesize DNA in the lab. A single-stranded DNA molecule, such as it shows, will serve as the template for the reaction. This DNA template is mixed with the DNA polymerase 3 holoenzyme, a primer with a sequence such, nucleotides see below, and a buffer containing magnesium, which is a cofactor necessary for the activity. So then we decide to perform our synthesis reactions as shown through A, part D. Then we determine the sequences of the DNA strands synthesized if we have the nucleotides described as in parts A through D in our experiment. Assume that if the DNA polymerase cannot incorporate a nucleotide into the nucleic acid strand, the reaction will terminate after the last incorporated nucleotide. Please do not include the sequence of the primer in your answer. Okay, so we know that the primer is going to match the 3' end to the 5' end of the primer. Okay, so the 5' end of the primer will match up to the 3' end of the sequence and follow backwards. Okay, so going from the right to the left on the sequence, we will find the 11th residue, and that will be where we will begin are synthesizing for parts A through D. So in part A, we have the 4 nucleotides shown. So because we have all 4 of those nucleotides in part A, we have everything we need to synthesize the entire rest of the strand. So continuing off of what the primer gave us, so starting with the 11th one from the right on the sequence, which is the T up there, you just go follow from right to left and read it, and that's what we get for part A. Okay. So then for part B, we have everything we need except the only difference is we have a DCMP, which is a deoxycytomonophosphate, something like that. So because we only have the monophosphate, we don't have enough phosphates for us to be able to synthesize the rest of the cytosines. And so when we start, we only get to that first piece because we can't synthesize that next C. Okay, so then for part C, we have everything we need except we have a UTP, which isn't what we need to synthesize is it thymine? Is that what it's called? Thymine? I always want to call it something else. Yeah, so we can't synthesize the thymine, so we only can do the first 2, the A, C, but we can't do the third, which is a T. So for part D, we have everything we need except what we need to synthesize the G correctly. It's a guanosine triphosphate, but not a deoxyguanosine triphosphate. So because it's not incorporated according to what we need for DNA, we can only get those first 3 as shown in part D. Okay, so then on to question 5. It just says below is a list of proteins involved in DNA replication. And then part A asks which of the above proteins is found in prokaryotes. So as shown, we've listed DNA gyrase, the TOS DNA primase, DNA polymerase 1 and 3 alpha subunit. Okay, so those are the ones that are involved in prokaryotes. For part B, it asks which are found in the mouse musculus, which we found to be CDCB, DNA polymerase E, DNA primase, DNA polymerase alpha, telomerase, pCNA, and MCM helicase. And then for question C, it asks which is involved in chromosome compaction. That's just the DNA gyrase. For D, it asks which is involved in the initiation stage of DNA replication. That's the CDC6. Is that what it is?
Yeah.
Okay, and then the DNA A. Okay, and then for the last question, it asks which of the above proteins is involved in the termination stage of DNA replication. That's the TOS and the MCM helicase.
Thanks for watching.